Sorry, little thing is spinning. I don't have confirmation that we're recording yet. Okay, now it tells me that we are recording. So here we are. Let's get back to okay, let's get back to not that. Come on, Wagner. Okay, let's get back to the presentation. Can everybody see this now? I'll take that as a yes. Okay, I got to meet this lady a couple of times before she passed. This is Lynn Margulis. She got her degree from Berkeley, spent part of her career at Boston University, but most of her career at the University of Massachusetts at Amherst. And she's a name you really ought to know because in 1970, under her married name of Lynn Sagan, she published a book called Origin of Eukaryotic Cells, and it transformed biology. Now, like everybody, she stood on the shoulders of people who had come before. So the book took up some ideas from some earlier 20th century biologists, like an American guy named Ivan Wallin and a Russian by the name of Mietskodsky. But she was able to back these up with data from this brand new hot field called molecular biology. Right, this is 1970. The double helix has only been worked out 16 years before. All of the classic experiments have been done that are working out transcription and translation and all of that. And there's this whole new flood of data coming in that would have been unimaginable 20 years before. And the field of molecular biology as a field is starting to take shape. And then Lynn Margulis is able to tap into this and use some data that's coming in a new way. And she developed what's called serial endosymbiotic theory. And you ought to know this already, especially if you have ever spent more than 2 minutes in the company of Dr. Naylor, because she works with this. But for those who don't, she proposed that eukaryotes arose not as single organisms, but more like collectives. That specifically the mitochondria of eukaryotes. You do remember mitochondria, right? They're the powerhouse of the cell, which is apparently funny because of some meme or something like that. They're descended from free-living bacteria that entered an early eukaryotic cell. Maybe the early eukaryote was trying to swallow the bacteria and eat them. Maybe the bacteria were being parasites within the eukaryote. It almost doesn't matter exactly how it happened, but they took up residence inside the host and developed a mutualistic symbiosis. In other words, each one became, over time, dependent on the other, so dependent that now they cannot live without each other. Mitochondria can't survive very long outside of a eukaryotic cell, and most eukaryotes are absolutely dependent on their mitochondria. You wouldn't last 10 seconds if your mitochondria suddenly disappeared. They generate most of the ATP that you use. This was also true for eukaryote chloroplasts, which are descended from free-living photosynthetic bacteria, very closely related to modern-day blue-green bacteria, that also entered some early eukaryotic cells, whether they were eaten by an early eukaryote and failed to get digested, or what it was, we don't know. Well, I think we do know, but it's not critical here. What is critical is that they formed a mutualistic symbiosis inside eukaryotes. Endo means inside, of course, right? An endoscope is an instrument that doctors use to look inside your digestive tract. And then symbiosis is Greek for together living. So living together inside, endosymbiotic. She later extended this and proposed that eukaryote flagella and cilia are actually descended from free-living bacteria, specifically a group called the spirochetes, that attached to the outside of host eukaryotes and formed yet another mutualistic symbiosis, that early eukaryotes were able to swim because they had bacteria stuck on the outside. And eventually, all of these endosymbionts lost their ability to live independently and became so tightly integrated into the overall, life of the cell that we no longer used to thinking of them as ever having been independent organisms. But the clues, says Margulis, are still there. What are those? Well, for one, mitochondria and chloroplasts have their own DNA. We can sequence DNA now. We've been able to do that relatively cheaply and easily ever since the early 90s. And mitochondria and chloroplast DNA sequences, when you compare them with other organisms, they're more similar to the DNA of bacteria than they are similar to host DNA. Now, you have to be careful there, because one of the fun things that's happened is that most of the genome of mitochondria and chloroplasts has actually been transferred to the nucleus. You've got genes in your own nuclear DNA that ultimately came from bacteria. You're part bacteria, man. We've all been genetically engineered. Ugh. But mitochondria and chloroplasts still retain their own DNA. They have to have it. They can't live without it. It encodes things like their transfer RNA molecules that they need. And their DNA is usually circular, not linear, right? Your chromosomes are basically long strips of DNA with a beginning and an end. Bacteria have DNA that's in loops, right? One big loop and then a bunch of smaller loops called plasmids. Mitochondria and chloroplasts have circular DNA, loopy DNA, which is a bacterial trait, not a eukaryote nuclear trait. Mitochondria and chloroplasts have their own ribosomes. Generally, they're the same size as bacterial ribosomes, not eukaryote ribosomes. You measure the size of a ribosome by something called the Svedberg unit, or a written capital S. And a Svedberg unit has to do with how fast things settle in an ultracentrifuge, because Dr. Svedberg was the guy who invented the ultracentrifuge. And it's a function of both size and shape. We don't really have to know how they work. But bacteria typically have ribosomes that are 70 Svedberg units, and eukaryote cytoplasm has ribosomes that are 80 Svedberg units. And mitochondria and chloroplasts typically have bacterial-sized ribosomes and bacterial-sized ribosomal RNAs. Mitochondria and chloroplast also share with bacteria the initiation of translation with formal methionine instead of methionine. You might have learned that translation always begins with the amino acid methionine. Maybe you file that away somewhere in your brains, but yeah, translation always begins with a methionine and a codon AUG on the messenger RNA. Bacteria and mitochondria and chloroplast always start with formal methionine, slightly different version of that amino acid. That happens to be a map of the human mitochondrial genome. You don't need to worry about the details, except that, first of all, it's a loop. Second of all, you can see mitochondrial genes for things like transfer RNAs. If you can see them, they're in fairly small print in ordinary type instead of in bold. Like up close to the top, there's something that says tRNA, PHE, and tRNA, THR, and so on. That's the sites of mitochondrial RNA, genes for mitochondrial transfer RNAs. Ribosomes are made up of proteins plus other RNAs. You have ribosomes that contain 28S and 18S RNAs. That's that Svedberg unit again. Prokaryote ribosomes have 16 and 12S sized RNAs in them, and so do mitochondria. And then, of course, the whole thing's a loop. Mitochondria and chloroplast are also surrounded by a double layer of membrane. You can see that right there. There's a cross-section through a mitochondria with those little parallel folds inside, the mitochondrial cristae. And there's a double layer of membrane surrounding this thing. And that makes sense if you assume that at one time, mitochondria and chloroplast ancestors were engulfed by a host. So the original membrane of the ancestor would be the inner membrane, and then part of the host membrane would go on and form that outer membrane. So structurally, mitochondria and chloroplasts, but not other things like, Golgi or things like that. Structurally, that's consistent with endosymbiosis. There's a diagram from a paper she came out with in 1990. And if she's right, then at some level, we have to replace our metaphor of the tree of life with something more like a network of life. Branches don't only divide, coming from common ancestors. They don't only split. They also diverge. All right. They diverge, they split, but they also can make like John Lennon and come together right now. Over me. Sorry. Branches can come together, they can join, and our last common eukaryote ancestor is actually a grouping together of at least 3, and in her hypothesis as of 1990, 4 different lineages of microbe. One group of bacteria that was ancestral to eukaryote cytoplasm and nucleus, and one that was ancestral to the mitochondria, and one that's ancestral to chloroplasts, and as she thought at the time, one that was ancestral to the cilia and the flagella. Eukaryotes are not single organisms. They're collectives of different organisms, in a sense. And she's basically right about this, although, as I've hinted, the idea of cilia and flagella being descended from symbionts is currently not really accepted, although there was reason for her to come up with this. This was not an idea that she pulled out of nowhere. But she's basically right about mitochondria and chloroplasts and lots of other examples as well. Endosymbiosis is all over the freaking map in the living world, where 2 unrelated organisms will come together and one may penetrate inside the other, but they'll end up forming an alliance for mutual benefit. The trees in my front yard are growing as well as they are because they've got fungi growing into their roots, taking nutrients from them, but supplying minerals to their plant hosts. They're called endomycorrhizae. You can buy endomycorrhizae spores and inoculate your garden or your potted plants with them. We make use of this all the time. There are lichens growing on the trees in my front yard, and those are symbioses between green algae, protists, and fungi. There are more E. coli bacteria living in your intestine than there are humans on the planet, and you would not be healthy if you didn't have them. In fact, there is new evidence that the bacteria living inside your gut exert an enormous impact on your own health. And even though I can't guarantee that all of them work, you can now buy foods with bacteria in them that are supposedly will improve your health by replacing, poor quality bacteria in your gut with good bacteria. If you've ever bought something that included probiotics, that's what those are. You're eating symbiotic bacteria. So endosymbiosis is absolutely everywhere, and Margulis drew attention to it and worked out ways in which it's absolutely fundamental to understanding eukaryote evolution. In particular, there's lots of single-celled eukaryotes that contain endosymbionts. And these are not identical with the first eukaryotes. The first eukaryotes would have lived over 2 billion years ago and have long been replaced by our current biodiversity. But they can give us some insights into how endosymbiosis might have looked in the very first eukaryotes of all. Just like I could go study the modern Amish to understand how my ancestors made a living 200 years ago, I can go to some of these modern eukaryotes to get an idea of how these early symbiotic associations might have worked. So, for example, that's a giant freshwater amoeba called Pelomyxa palustris. Tends to like living in muddy substrates with not much oxygen. These probably are found on the bottom of Lake Conway, for example. They can get up to 5 millimeters long. These are not quite that big, but they can get absolutely huge. They don't have any mitochondria, but some of those granules that you see in the cytoplasm are actually bacteria, and those bacteria carry out respiration for it. They're much more independent and much less structurally reduced than mitochondria, but they do the same thing as mitochondria. So Pelomyxa can give us some insight into what early stages of the mitochondrial symbiosis might have looked like. These guys you just might see pretty soon. This is a very common protist called paramecium. You might have seen some of these in 1441 because Dr. Carafit told me that he often orders these for his classes. And individuals in this species can be packed with symbiotic bacteria. The symbionts, by the way, by convention, different types of symbiont are given the names of Greek letters. So these are called kappa particles and alpha particles and mu particles and things like that. What they are is symbiotic bacteria. And what these symbionts do is they produce toxins that kill other Paramecium aurelia that don't have those symbionts. So, yeah, it's a way of, I guess, reducing the competition in the pond, I suppose, is killing other Paramecium aurelia that don't have the same symbionts as you, which makes me cool. Well, okay, sorry, need more coffee. Which I think is funny because it's interesting to see that college students are not the only individuals that have rivalries based on what Greek letters they're carrying. Be funny if we had Paramecium that had pi, kappa, and alpha particles, having a rivalry with Paramecium that had sigma and nu particles or something like that. But yeah, Paramecium evidently has a Greek letter system, much as much as American college students do. Okay, here's another one. We'll cover these in a little bit more detail coming up, but amoebas are single-celled organisms that don't have a fixed shape, kind of like big blobs that ooze from place to place. Many amoebas are naked amoebas, but there's quite a lot that form shells around part of the cell, and then they stick little extensions of the cell out. Amoebas with shells are called testate amoebas. And we'll cover these guys again later. You don't have to memorize this just yet, but you will see these coming up. And this is a testate amoeba called Polynella chromatophora. The thing that looks like a big vase is the shell. Inside is where there's most of the amoeba. On the right, if you look very closely and maybe take drugs, you can see some very thin, fine extensions of the cell sticking out, almost like little tentacles. But you can see inside there's a pair of things that look like bluish-green horseshoes. Those are blue-green bacteria. Those are photosynthetic symbionts living inside Polynella. And Polynella is not thought to be especially closely related to any plants or any algae. So this is thought to be a case of convergent acquisition of a symbiont. The ancestors of Polynella somehow got blue-green bacteria living in them and have passed those on to their descendants independently of what plants and algae did. I didn't used to do this in our lab because they used to do it in 1440 or 41. I might try and reintroduce it. But termites, of course, feed on wood. But termites can't actually digest wood. What they depend on is a rich community of protists, single-celled eukaryotes living inside their guts. And this is a couple of one species of termite gut protist in genus Trichonympha. They're a lot of fun to watch. They've got this bulgy shape and lots of very elongated flagella up at the front, up around that kind of nose cone that you see at the top. They're actually kind of wobbly when they swim. They look a little bit like Santa Claus, shaking when they laugh like a bowl full of jelly. So these are symbionts inside termites that the termites depend on to break down particles of wood. But Trichonympha can't break down wood. What it's got is bacteria living inside it, and those break down tiny particles of wood. In the lower half of the cell, the posterior half, you can see some kind of irregular, little lumpy things inside. That's particles of wood, and those are getting broken down by the Trichonympha, those are getting broken down by the bacteria, which live inside the Trichonympha, which live inside the termites, which live inside the apartment I lived in my senior year. So yeah, you have life as kind of a set of Russian dolls with symbiont living inside symbiont. The photo on the right, you can see this hairy stuff coming off the posterior half of Trichonympha, the bottom half, and those are bacteria that are attached to the outside. So remember I mentioned that Margulis had this idea that cilia and flagella were descended from bacterial symbionts. That idea has not really withstood scrutiny, but you can see where she might have gotten the idea. They're about the same size as these bacteria that can live on the exterior of some protists. I'm imagining you all going, ooh, ah, right about now. Okay, these guys are the cause of a human condition. This is Trichomonas vaginalis. They're protists. They live in the human vagina. I used to see these guys when I used to work at the Berkeley Free Clinic back in my freewheeling grad school days. I was a lab tech, and I'd look at vaginal samples, and sometimes I'd spot these. And in low numbers, they're pretty normal. If there's not many of them, they don't tend to cause problems. If they overgrow and you get a lot of them, they cause a vaginal infection that is kind of miserable as far as I can tell. And that's one of the things that we would treat. And anyway, I bring this up because these are anaerobic protists. They are killed by oxygen. The vagina is normally an anaerobic environment. And in fact, you've got lots of little anaerobic pockets in your body. Much of your gut, parts of your mouth, the little pockets between your teeth and your gums, those can all be mostly anaerobic almost all of the time. And Trichomonas doesn't have mitochondria, as you might expect, given that it's anaerobic. And protists like Trichomonas used to be, we thought that they branched off the tree before any eukaryotes acquired mitochondria. We thought these were eukaryotes descended from ancestors that lived before that symbiosis ever took place. However, that's a slice through Trichomonas, and if you can see, there's a bunch of gray dots in the middle of Trichomonas that have been labeled with a capital H. Those are organelles called hydrogenosomes. The hydrogenosomes actually break down, I think they break down pyruvate and yield hydrogen as a byproduct. But they don't carry out oxidative respiration anymore. They don't have any genome left, but on other points, they're very similar to mitochondria. What they probably are is former mitochondria that have been repurposed and have lost their genome completely. And in fact, in Trichomonas' DNA, there are genes that are now in the nucleus, but they're most similar to mitochondrial genes. So they are former mitochondrial genes that ended up moving into the nucleus. This sort of gene transfer actually happens a lot more commonly than sometimes we think. It's not that uncommon in evolution for DNA from one source to end up getting spliced into the genome of somebody else. It's basically natural genetic engineering, and this is one of the cases where it's happened. So we now think that even eukaryotes that don't have mitochondria are descended from ancestors that did, and they've lost them secondarily. So the ancestors of Trichomonas used to have functional mitochondria. We now know, and we'll talk about these towards the end of the course, there are even some animals that have ditched their mitochondria completely. Nothing you're likely to have seen. They're microscopic, but they're completely anaerobic. They live without mitochondria, and yet there's evidence that they used to have them. So there's only one eukaryote known so far that's completely ditched its mitochondria, which you just might spot not in today's lab, but maybe in Thursday's lab. All other eukaryotes either have mitochondria or they have organelles that are descended from mitochondria, like those hydrogenosomes. But even the ancestors of this guy still did have mitochondria. So having mitochondria is fundamental to being a eukaryote. And according to a hypothesis, mitochondria got acquired when little blobs or blebs emerge from a host prokaryote, engulf these protomitochondria, and as those blebs grow and wrap around mitochondria, they not only create these vacuoles where the mitochondria live, but it also created the endomembrane system, right? The endoplasmic reticulum and the Golgi body and all of that, and created the nuclear membrane as well. So it would have been the acquisition of mitochondria that is ultimately responsible for creating the entire architecture of a eukaryotic cell, not just the mitochondria themselves, but the endomembrane system and the nuclear membrane as well, all ultimately result from this. Is that not, I ask you, freaking cool? I will assume you're all saying yes, Dr. Wagner. And there's actually some archaea today. Archaea are thought to be the closest relatives to the nuclear genome of eukaryotes. These are a couple of them. These things labeled ARCH, Giganthalma caracense, and they form blebs, and these attach to bacterial endosymbionts. So you have little bacteria that are attaching to the outside of archaea like this, and this could be a model for how early eukaryote evolution looked. And there's a paper you can look up if you want to track this down further. Other models propose that there are the early ancestors of mitochondria penetrated into early hosts. And there are some living bacteria that do this as well. Over there on the left, you can see this thing. This is a cross-section through a large bacterium. And there's a small bacterium that looks a little bit like an infinity sign, and it's darker inside the larger bacterium. This is a bacterium that's a parasite on other bacteria. It's called Daptobacter. So maybe this is closer to the model of how mitochondria were acquired. There's still plenty of work to be done on the question. We don't have all of the details worked out yet, but what we can get is models for how it happened. We can find these living taxa that show you what life is capable of and give you models for pathways by which ancient life could have done it. In addition to Daptobacter, we have other bacteria that we know are obligate parasites inside eukaryote cells. This is a couple called Rickettsias. Rickettsias, there are some that cause human diseases like typhus. They lack cell walls completely, and they're obligate intracellular parasites of eukaryotes. So maybe this is closer to what the earliest mitochondria looked like. And in fact, if you analyze complete DNA and protein sequence data, you get this tree. These are all bacterial sequences, but the ones indicated with those red lines are Rickettsia bacteria, these cell-less intracellular parasites. And the ones indicated with the blue bar, those are mitochondria. So mitochondrial DNA is most closely related, not just to bacteria, but specifically to Rickettsias. So Rickettsias are the closest relatives, we think, of the lineage of bacteria that ultimately evolved into mitochondria. How would you go from being a parasite to a symbiont? That question got asked beginning in 1966 by Dr. Kwang John at the University of Tennessee. And this is the big blobby thing that you're seeing on the left is an amoeba in the genus Amoeba. There's actually a bunch of amoeba genera. This one, I think, is species Amoeba proteus, and it's pretty large. They get up to 0.5 millimeter or sometimes more. I don't know if you've seen these guys before in maybe 1440 or 1441, but they're easy to raise and they're popular in labs. And we would probably be looking at some under different circumstances. And Dr. John succeeded in infecting his amoebas with bacteria. He found a strain of gram-negative rod-shaped bacteria that could enter amoeba cells and live inside and parasitize the amoebas, drawing off energy and molecules from the amoeba's own metabolism. So yeah, kind of strange to think of amoebas getting sick. And in fact, most of his amoebas died, but a few survived. A few, for whatever reason, were just a bit resistant to these bacterial infections. And so he cultured the survivors, and every time an amoeba reproduced by dividing in two, infective bacteria would go along for the ride into the daughter amoebas. So these infective bacteria got passed on from one generation to the next. And at first, Dr. John found that he could cure infected amoebas by giving them a dose of antibiotics. He could kill the bacteria, the amoebas would be unharmed, and they would just go on and live as they normally would. But after about 5 years, the surviving amoebas had become so dependent on the bacteria living inside them that they could no longer survive without them. If he zapped these amoebas with antibiotics, it killed them. It didn't cure them. And the reason was that bacteria were now making a key enzyme that infected amoebas were no longer able to make. They'd lost the ability to make this on their own. So the details are a little bit different, but you can see that with just a bit of selection, it's possible to go pretty quickly, 5 years, from a bacterial parasite to a bacterial mutualistic symbiont. And he's refined this over all these years as a laboratory model for how the first eukaryote endosymbionts might have been acquired. If nothing else, it gives you proof of concept. It shows that it's possible for a bacterial parasite to become a bacterial mutualist. Okay, right. Before I go on, I'll give you guys another stretch break in just a second, but one little thing that it occurs to me ought to be said. I try not to get political in classes like this. It's not really my job. It's probably something you'd rather not hear about, especially because these days it's so damn depressing. And I certainly can't fault you for that. But one little thing that it's good to remember. Darwin didn't actually say it, but he's often associated with the phrase survival of the fittest. Although he did not say that, that was a guy named Herbert Spencer, who was kind of a boring dude. And that's associated with a model of evolution that is hyper-focused on ruthless competition. Survival of the fittest means that only the meanest and fiercest and most badass can survive. And people hear this and they think about, I don't know, some savage shark fiercely tearing apart a helpless, unfit mackerel on Shark Week on Animal Planet or whatever channel that is. Or, a pack of savage lions tearing apart, a gazelle that just couldn't run fast enough on the Serengeti Plain, or whatever kind of, big animal porn they've got showing on Animal Planet or whatever it is. And that's what people tend to think of when they think of evolution, that it's all about competition, and often violent competition where only the strong will survive. I'm not saying that never happens. There's a reason why nature is called a mother, because Mother Nature can be one mean mother, let me tell you. But very, 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 very often, evolution is all about not who is the most fierce competitor, but who's the best collaborator? This endosymbiosis type thing will be finding again and again. Every organism that's ever been looked at in any kind of detail has symbionts in it. You could argue there is no such thing as a single organism, that in some degree, in some measure, we're all collectives. And one of the things that Lynn Margulis was always very insistent on is that cooperation works much more often and much better in the long run than that kind of mindless competition that I've talked about. That life does great things when it comes together. And if you're feeling depressed or concerned or saddened by the rather depressing scenes that are coming out of our cities, or hell, for that matter, in Conway last night, that might give you a certain degree of comfort. If you're feeling like the world right now is all about survival of the fittest, very often that means survival of the most cooperative. And I'll go ahead and leave it at that, and I'll pause recording right now. And after another stretch and coffee break, we'll pick this up again.